Hi everyone, today I'm going to explain you about the breast histology. Normally the breast contains two types of cells. One is luminal cells and another one is myoepithelial cells. And these are the normal luminal cells, which are we also known as albuminal cells. Why they are luminal cells? Because these cells are located towards the lumen of the breast. And another is myoepithelial cells, which are located around the lumen of the cells. That means far away from the lumen of the cell. Away from the lumen we call our myoepithelial cells, towards the lumen we call them as luminal cells. So they has and not only that, there is a normal breast has rule of 2. That is, rule of 2 is nothing but there are two types of epithelial cells. One is luminal and another is myoepithelial, and another is two types of stroma the breast contains. One is interlobular stroma and another one is intralobular stroma. And we have two duct structures. One is large ducts and terminal duct unit. So this rule of 2 applies to the normal breast. So whenever there is a cancer, all these structures will lost. Especially in the cancer patients, you will not see this myoepithelial markers. These myoepithelial markers will be negative in the cancer patients. So you are supposed to know what are the markers. Why it is important? Because myoepithelial markers are surrounding the outer layer. So whenever there is a cancer, these markers will be absent, because the cancer cells, to get into the stroma or to the spread, they're supposed to break the basement membrane. These myoepithelial cells will be lining the basement membrane. So they break the basement membrane, they enter into the stroma. Okay, so what are the myoepithelial markers you are supposed to know? Because whenever you are suspecting invasive carcinoma of the breast, whether it might be lobular or ductal, this epithelial markers will be absent, especially P63, P40, calponin, SMA, SMMH, CK5/6 and S100. So these markers can be nuclear, cytoplasmic, or both nuclear and cytoplasmic. Same like myoepithelial, we have luminal markers, which are CK19. These luminal markers are helpful for us to identify whether the primary origin of the tumor, whether the primary origin is from the breast itself or from the metastasis. So what are the luminal markers? We have CK19, EMA, E-cadherin, BCL2, GATA3, mammoglobin, GCDFP, CK7, CK8. Among all the markers, GATA3 plays a very important role, because whenever there is a primary breast cancer which is metastasizing to other areas, so of the other areas of the body, the primary, to identify the primary origin of the tumor, we use GATA3, especially in case of metastasis. If GATA3 is not available, we have another marker, that is NYBR, we use to identify the primary. And third and last is AR and ER is used. So very important is GATA3, because it is useful to identify the primary in case of metastasis, because it helps to identify these are the breast luminal cells which are proliferating due to metastasis. So these luminal markers are very useful in identification of primary, whereas myoepithelial markers are helpful because they are always absent in invasive cancer of the breast.